Welcome to this lecture on the basics of neuroscience. Today, we are diving into the electroencephalogram, commonly known as the EEG. The human brain is a complex organ, constantly buzzing with electrical activity. But how do we actually measure and understand this invisible energy without opening the skull? By the end of this video, you will understand not only the biology behind brainwaves, but also the mathematics we use to model them. Let us begin our journey into the electrical life of the mind. To understand how we measure brain activity, imagine standing outside a massive football stadium with 80,000 cheering fans. You cannot possibly hear what one specific person is saying. However, by listening to the overall roar of the crowd, you can tell exactly what is happening. If the crowd suddenly erupts into a massive synchronized cheer, you know a goal was just scored. A low, steady murmur means the game is likely paused. The human brain works in a very similar way. It contains billions of neurons, acting like those individual fans. An EEG is essentially a highly sensitive microphone placed outside the stadium of your skull to listen to the collective electrical roar of your neurons. Now that we have our stadium analogy, let us look at the formal definition. An electroencephalogram is a non-invasive medical and research technique used to measure and record the macroscopic electrical activity of the brain over time. Because the skull blocks a lot of the signal, we place highly sensitive metal discs, called electrodes, directly onto the surface of the scalp using a conductive gel. These electrodes act as our microphones. The EEG machine then takes these microscopic, incredibly weak electrical signals and amplifies them. It plots these voltage fluctuations over time as continuous wavy lines on a computer screen, allowing us to analyze the rhythm and volume of the brain's electrical noise. Understanding the equipment is only half the story. We must also understand the biology. Where exactly does this electrical signal come from? The EEG does not measure single action potentials. Instead, it detects the tiny electrical charges that result from the activity of thousands of neurons firing together in perfect synchrony. Specifically, it captures the postsynaptic potentials of pyramidal neurons, which are located in the outer layer of the brain, known as the cerebral cortex. When these neurons fire together, they create voltage fluctuations that we can measure. By carefully analyzing the frequency and amplitude of these resulting brainwaves, neuroscientists can figure out exactly what state your brain is in. So, why is measuring this electrical activity so important? The EEG is an incredibly powerful tool because it provides a real-time window into the living, functioning brain without requiring any dangerous surgery. Its most famous and critical real-world application is in the diagnosis and monitoring of epilepsy, a condition where the brain's electrical activity becomes abnormally synchronized, causing seizures. Furthermore, it is the absolute gold standard for studying sleep, allowing scientists to map out different stages like REM and deep slow-wave sleep. It is also routinely used in emergency rooms to determine brain death in comatose patients and in cognitive research to understand how we process language and emotions. Before we dive into the mathematics, we need to establish how neurologists categorize these brainwaves. Brainwaves are grouped into standard frequency bands, measured in hertz, which means cycles per second. At the slowest end, we have delta waves, ranging from 1 to 4 hertz, which occur during deep sleep. Next are theta waves, from 4 to 8 hertz. Then we have alpha waves, ranging from 8 to 13 hertz. Alpha waves are universally known as the dominant brain rhythm when a person is awake, completely relaxed, and has their eyes closed. Finally, beta waves range from 13 to 30 hertz, indicating a highly alert and focused brain state. Now that we know the frequency bands, let us look at how we actually calculate frequency from raw EEG data. The fundamental mathematical relationship we use defines frequency as the reciprocal of the period. The formula is F equals 1 divided by T. In this equation, the symbol F represents the frequency, measured in hertz, which tells us the number of cycles per second. The symbol T represents the period, which is the total time required for one complete wave cycle to finish, measured in seconds. If the period T decreases, meaning the wave cycles finish faster, the frequency F increases, indicating a more alert brain state. Let us apply this formula to a real clinical scenario. A doctor is reviewing an EEG of a patient resting comfortably with their eyes closed. The doctor measures the time for one complete brainwave cycle and finds the period is exactly 0.1 seconds. We need to calculate the frequency. We start with our formula, F equals 1 divided by T. We substitute the given period of 0.1 seconds directly into the denominator. Dividing 1 by 0.1 gives us exactly 10 hertz. Looking back at our standard bands, 10 hertz falls perfectly into the middle of the alpha band. This makes perfect biological sense, as the patient is awake, relaxed, and has their eyes closed. Beyond just counting cycles, scientists often model the actual shape of the brain waves using mathematical functions. The most basic way to represent a rhythmic, synchronized brainwave is by using a standard sinusoidal wave equation. The formula is V of t equals A times the sine of 2 pi ft. Here, V of t represents the instantaneous voltage of the EEG signal at any given time t, usually measured in microvolts. The symbol A stands for the amplitude, which tells us how tall the wave is. This physically reflects how many thousands of neurons are firing together in perfect sync. If the amplitude increases, the waves become taller, which typically happens during deep sleep. Let us see this sinusoidal model in action. During a sleep study, a patient enters deep non-REM sleep, producing slow delta waves. We are given an amplitude of 50 microvolts, a frequency of 2 hertz, and we want to find the voltage at exactly 0.125 seconds. We substitute these three values into our equation. Multiplying the terms inside the sine function gives us 2 times 2 times 0.125, which equals 0.5. We are evaluating the sine of 0.5 pi radians, which is equivalent to 90 degrees. The sine of 90 degrees is exactly 1, representing the absolute peak of the wave. Multiplying 50 by 1 gives a final instantaneous voltage of 50 microvolts, meaning the wave has reached its maximum positive peak. Having calculated both frequency and voltage, let us now visually compare these brainwaves. On screen, you are looking at 4 distinct EEG traces, each representing 1 second of brain activity. Notice the top trace, the beta wave. It is very fast and tightly packed with a low amplitude, representing an active, busy mind. Just below it is the alpha wave, which is slightly slower and more rhythmic. As we move down to the theta and finally the delta wave at the bottom, you can clearly see the physical changes we discussed. The frequency decreases, meaning fewer cycles per second, but the amplitude drastically increases. The delta wave is tall and slow, perfectly illustrating the massive, synchronized neuronal firing that occurs during deep sleep. We have covered a lot of ground today, so let us review our key takeaways. First, we learned that the EEG is a vital, non-invasive tool that measures the macroscopic electrical activity of millions of synchronized neurons in the cerebral cortex. Second, we saw how this tool is absolutely crucial in medicine and research, from diagnosing epilepsy to mapping the stages of sleep. Third, we explored how brain states are categorized into specific frequency bands, such as delta, theta, alpha, and beta. Finally, we successfully applied mathematical models using the relationship between frequency and period, as well as sinusoidal functions, to quantify these invisible waves. You now have a solid foundational understanding of the electroencephalogram.